Uh, Hello, everyone. Thank you for joining us for today's Molecular Connectivity Online Series, or MCOS. I'm Cindy from New Jersey Institute of Technology. In case you are new here, the Molecular Connectivity Working Group is a group of researchers that aim to advance molecular imaging as a tool to study brain connectivity. We organize this online series as a platform to exchange ideas and promote scientific research on molecular connectivity. You can follow us on Twitter and LinkedIn, and also go to our website and subscribe to our newsletters. Today, I'm happy to introduce the speakers, Dr. Mattia Veronese and Mario Severino. Mattia Veronese is an associate professor in biomedical engineering at the Department of Information Engineering, University of Padua, and an honorary senior lecturer in neuroimaging at King's College London. He is a biomedical engineer by training and holds a PhD in PET kinetic modeling. His main research interest is related to the development and validation of molecular imaging, biomarkers, and their use for drug development and precision medicine. Mario Severino is a PhD student in biomedical engineering at the Department of Information Engineering, University of Padua. He holds a master's degree in biomedical engineering. His primary research focus is on applying PET molecular connectivity and network science approaches, aiming to establish these methods as the precision medicine biomarkers. In today's talk, they will talk about molecular connectivity in neurotransmissions. I would also like to introduce Dr. Carlo Cavalieri. He's a MD PhD in SDN Research Institute for Diagnosis and Nuclear Medicine. His research focuses on multimodal neuroimaging biomarkers of neurodegenerative disorders, cognitive declines, and disorders of consciousness. Once again, thank you for coming. Thank you for coming, and let's welcome Dr. Veronese and Mario for today's talk.
Thanks, Sing. I'm going to share my screen. That is okay. Can everyone see it? Okay, super.
Yes.
First of all, I want us to thank the group for the invitation. So most importantly, all the friends I've seen here connected to these presentations. I hope this will be a nice sail towards the weekend and not much of a waste of your time. So we start today talking about molecular connectivity neurotransmissions, but as an engineer, I cannot start by praising the wonders of the brains. And this is kind of like immediate when you start to look at the brains in numbers. So brains is a structure that more or less weighs a little bit more than 1 kilo. It occupies a volume of 1.5 liters. But inside this 1.5 liters, more or less, is the content of a plastic bottle, a water bottle. You have 86 billion of neurons and more or less the same number of glial cells. What is very interesting is like these neurons and cells makes together a very complicated network. So you have a possibility in every brain. Okay, that's the wonder of it. There is a possibility for 200 trillion synaptic connections. Just to give you a comparison, the Milky Way contains more or less between 200 and 400 billion stars. So in every brain, in every human brain, there is a complexity that is much bigger than what is the galaxy. So, looking at the brain as with the same wonder of the universe. Also, brain is a very sophisticated machine. It can process 10 trillion operations per second.
Mattia, sorry,
yes.
Are you going forward with the slides? Because we look only the first one again.
I'm going through the slides. Can you see that I'm moving the slides?
No.
Only the first one.
Let me just share it again. Okay.
Sorry.
No, it's okay. Actually, thanks for telling me at the very beginning. Can you see it now? Can you see my slide moving?
It's opening, I think.
No, it's a black screen and
Yeah.
Maybe you want Mario try to share it?
Yes, I will share.
Yep, got it.
Okay.
Thanks, Chris.
This is the correct slide.
I still see like a black screen on my side.
I can see a new slide.
I can see the slide. I can read it for you if you want.
Okay, so I can go by heart. So I think we are in the second slide, no?
Yes.
Mario? Okay, super. So, I mean, what I'm just saying is like, I'm praising the brains as a complexity here because, you know, it has some fantastic performances. And it's even more fantastic if you think that at the end of the day, the brain is made by lipids and water for majority of its components, but it's powered by electrical signal. And at the very core of this electrical signal, there is neurotransmission, which is a fundamental aspect of how the brain works. Now, if you have the diagram,
Firstly, we can see either Mattia or the slides, so not both.
okay. Can you go to the next slide?
Yes.
Okay. So I mean, it's not a place where we're going to discuss neurotransmissions here, in the sense that everyone here is somehow related to neuroscience and know this process very well. You know, this is basic biology. But you know, in neurotransmissions, we have the pillars of how the brain works and how the communication between neurons works. And as you know, there are multiple aspects to account in neurotransmissions. Starts from the synthesis of neurotransmitters up to the productions and storage of neurotransmitters at the synaptic terminals, the release that is very, very regulated, as well as the uptake and the neurotransmission binding to neuroreceptors. Now, what is important is like we can image neurotransmissions with molecular imaging. And if you, Mario, you're keen to go to the next slide, we know that when using PET and SPECT imaging, we can detect and quantify different aspects of neurotransmission. So we can start from neurotransmitter synthesis. I'm thinking, for example, the case of fluorodopa, where you have a conversion rate from dopa into dopamine. We can measure the density of vesicles. We can measure, and therefore, you know, the amount of neurotransmitters that is available at release. We can measure receptor density as well as neurotransmitter transporter density. And by doing competitive studies with blocking or challenges, we can even measure neurotransmissions release. Now, this capacity of doing this kind of imaging in vivo, okay, while the brain is functioning, has led us over the last 40 years to fantastic discovery, not just how the brain works in normal physiological conditions, but also to allow us to identify mechanisms of brain disorders, supporting drug developments. If you think that in the 90s, a lot of pharmaceutical companies, they have their own molecular imaging facilities, as well as definitions of clinical biomarker. I'm thinking, for example, here the case of DAT scans for Parkinson's disease. Now, if Mari go to the next slide, just make a point here. So if you have work with imaging of neurotransmission system of any type, you know that traditional methods focus the analysis of tracer-specific binding in a predefined list of regions. Generally, these are region of interest, anatomical regions where you have the highest tracer activity, which generally correspond to, you know, the hub of the key regions for that particular neurotransmission systems. And when you work in PET quantifications particularly, we have this hypothesis of independence. So we tend to characterize each region one at a time, ignoring that, you know, these regions are all connected to each other. Now, there are two important limitations behind this assumption. The first is like we are using an univariate approach for a system that is complex and multivariate by definitions. All the different regions interact with each other. Okay, so it's very difficult. It's very difficult, and if you want, it's kind of niche to analyze one region at a time, because we somehow lose all the information around this complexity and to the between regions correlations. It also, I need to say that this view of going one region at a time, violates what is like the modern view that we have of the brain, where, the different brain regions are not the defined isolations, but all connected to each other. So we have like a network model of the brains, and I think everyone will agree that this is probably the most modern and probably also the best performing model when we look into the brain. Now, there is nothing wrong against traditional methods. They work for the majority of the cases and for the majority of applications. But I think if we want to progress our knowledge, if we want to go beyond our traditional analysis, we need to change the perspective. And I think one change of perspective is represented by molecular connectivity. Now, if you go to the next slide, Mario. So here I need to make a disclaimer before we go into fights and discussion. So this is not a presentation to define what molecular connectivity is. I simply took the definitions here of our consortium, which stands as molecular connectivity as the use of molecular imaging to study brain connectivity. And when I say brain connectivity, I also include neurotransmissions. Now, in the literature, until we will reach to an agreement or even a more refined definitions, molecular connectivity, unfortunately or luckily, whatever you want, whatever part you want to take, is used as more like as an umbrella term, and tends to describe statistic methods that account for statistical interdependency between regional measurements obtained with molecular imaging techniques. So in other words, if you can take your PET measures and build from these PET measures a PET agency matrix where you have as element of this matrix different brain regions, and as the link between these two regions, some measures of strength of associations as derived from the molecular imaging, then you're going to build a PET agency matrix that will translate into a molecular connectivity methods. So when we apply this kind of methodology, statistical methodologies, for radio tracers that were targeting neurotransmission system, that's the place where, the place where we put our work and the perspective we are going to discuss in this particular presentation. Now, the advantages of this perspective is like by using network approach, you have the two limitations I was mentioning before, removed, because this approach is intrinsically multivariate and also intrinsically connected, because every region can be linked to any other regions. Mario, can you go to the next slide? So when we start to work on this project, Mario and I, together with the rest of the molecular connectivity team, we start to wonder, what kind of methods of molecular connectivity methods, as defined as before, have been already used to investigate neurotransmissions. And there were many. I mean, even we did our analysis last year, so we found ourselves that over the years, there were several studies, more than one, what we thought actually, that look at these methodologies to investigate molecular imaging studies targeting neurotransmissions. So our questions when we approach our review were about, okay, what methods have been used in what kind of applications? But most importantly, we start to ask ourselves, okay, what does molecular connectivity bring to the table? What is the value of molecular connectivity or these kind of methodologies as compared to the traditional value analysis? And this is the kind of question that we all ask ourselves, because, if there is no value of any type, why should we bother to do this complicated analytical framework? Okay, I've seen a hand raised. I don't know if we can go ahead or you want to have the questions at the end, Carlo?
Probably at the end, because Marco raised the hand, but probably is an error. So we can go forward. Yeah.
Okay. So that's where we started. Okay, and I think from this point afterwards, I can let the pass the microphone, virtual microphone, to Mario, so he can tell you more of what we did.
Yes, thank you, Mattia, and thank you all for the invitation. And yes, to answer these questions, we performed this scoping review of the literature, where our aim was to answer those questions. And to do it, we followed the PRISMA statement, that is a series of guidelines to report and to perform a review in a rigorous way. We recorded our the article principally from free database, Embase, Medline, and Scopus, and we ran the search on July of 2023, and another run on April of 2024. Here I've reported the eligibility criteria. We specifically focused on that use PET and SPECT as molecular imaging. And we use keywords like connectivity, covariance, and network with the aim to find those studies that better fit the criteria of molecular connectivity. Here I've reported the flowchart that we followed to screen the paper. I will go quickly through it. So after retrieving all the paper that at first were 7,000, a lot of them were removed before screening because there were a lot of duplicates. Then we performed, all of this was performed in a platform called Covidence to track all our processes. And then we screened the paper first by title and then by abstract, and then a full text article were assessed for eligibility. At the end, 488 paper met eligibility criteria, and from these, we extracted a subset of 39 studies that focused explicitly on neurotransmission. So these were the subject of studies included in this scoping review. Now, here I've reported some summary information regarding the article. Firstly, they were categorized according to the target system, and the majority of paper 23 focused on dopamine system, followed by serotonin, opioid, muscarinic, synaptic density, and glutamate. While regarding the molecular connectivity methodology applied, the majority used the pairwise Pearson correlation, followed by interregional correlation, principal component analysis and its variant like SSMPCA, independent component analysis, and partial least square. Then a lot of paper used also other type of methodology, like a combination of these or other, let's say, non-conventional approaches. While regarding the cohorts of subjects included in this study, the majority of the study employed a healthy control cohort, while the most investigated disease was Parkinson's disease, followed by mild cognitive impairment, schizophrenia, dementia with the Lewy body, major depressive disorder, Alzheimer's disease, and then anxiety, epilepsy, cocaine use disorder, and attention deficit hyperactive disorder. So this was a summary, and since it would not be possible to go through all the article included in the review, I've decided to select 3 of them as a case study. And then after that, I will also show some notable findings of also the other article included in the review. So moving to the first case study, we have a work of Fazio et al. Of 2020, which targeted the serotonin system. In this case, the cohort under investigation was composed by 20 healthy control and 20 patients with Parkinson's disease, scanned both at baseline and follow-up. And this study is particularly interesting because it's a longitudinal study. The subjects were scanned to measure the serotonin transporter. Indeed, the aim of this work was to investigate the role of serotonin transporter in the brainstem nuclei and in relevant projection area in early non-depressed Parkinson's disease patients. The molecular connectivity analysis focused on the pairwise Pearson correlation. So matrices were constructed using Pearson correlation at different density threshold, and then they were binarized to extract the graph matrix. In this case, they used both global integration matrix and segregation matrix. We have average global efficiency and local efficiency that are two measures to evaluate the efficiency of information transmission in the network as a whole and at the local level. While regarding segregation metrics, they use transitivity, that is a measure similar to the clustering coefficient, that is the tendency of the network to form triangle, and modularity, that measure the strength of a network to form communities or module. They also employed univariate statistical analysis that was performed between the region, both cross-sectionally and longitudinally, and the same type of statistical analysis was also applied, but to network metrics, in this case using permutation on parametric test and confidence intervals. Moving to the results, they found no statistical significant difference in univariate analysis, both cross-sectionally and longitudinally. The same was also obtained with network measures at baseline, and the authors suggested that these may be linked to a relative preservation of the serotoninergic network in Parkinson's disease patients compared to healthy control, while at follow-up, patients showed a reduced average global efficiency and an increased transitivity, as we can see in this plot that I've reported here. And these suggested a significant change in both integration and segregation of the serotoninergic network. Furthermore, they also found differences in the modular organization. While at baseline they found only one modular structure, at follow-up there were two modules, one that included the Raphe nuclei and putamen, and the other one comprised cingulate and other subcortical regions. This suggested a progressive disconnection between the brainstem subcortical region and cingulate, and the author suggested that this may be explained by potentially to the axonal degeneration and the synaptic protein loss. So to summarize this study, the main two key findings are that the serotoninergic system might become involved in patients as the disease progresses, as they showed with the longitudinal analysis. And importantly, these findings was only captured by network measure and not with the univariate analysis. And the authors suggested that maybe these subtle changes are diluted in univariate analysis because of the variability across individuals. Now moving to the second case study. In this case, the target system is the dopamine one, and we have a study of Fu et al. Of 2019. In this case, the cohort under investigation was composed by 15 early Parkinson's disease patients, scanned with both vesicular monoamine transporter 2 and dopamine transporter. And this study is interesting because they proposed a novel approach called the joint pattern analysis, and it is also a dual tracer studies with the aim to explore the performance of this model in extracting common and unique information between these two radio tracers in early stages of PD. So the approach starts with a univariate analysis that was used to identify regions with a binding potential significantly greater than 0. So only those regions were used for the subsequent analysis that consisted in this joint pattern analysis that is a combination of canonical correlation and orthogonal signal correction. So they first started with two input matrices, one representing the DAT scan and the other representing the vesicular monoamine transporter radio tracer. And they first demeaned and whitened these matrices to obtain data that were uncorrelated, so that each column of the data was uncorrelated with the other columns. And they reduced the number of features, trying to maintain the majority of the variance. So they applied this canonical correlation to these matrices, and it is an approach that consists in maximizing the correlation between pair of these canonical variate while decreasing the interdependencies within the same data. The objective of this part was to extract the common information between the two radio tracers. And then they applied instead a lasso regression approach to obtain to regress these canonical variates from the original matrices and to obtain the regression coefficient that are called the canonical correlation analysis weights and the residuals. And they use this orthogonal signal correction to extract from the residual instead the unique information of the two radio tracer. So to sum up this part, they were able to extract the common information between the two radio tracer that was the information maximally correlated between the two matrices and the unique information that was taken from the residuals. And from this, they were able to construct spatial covariance pattern that decomposes the gradient of dopaminergic loss in orthogonal components. And after applying cross-validation and other non-parametric approach, they found three main components that were maximally correlated between the two radio tracers. The first component was linked to the disease-induced asymmetry between the less and more affected dorsal striatum. For example, I've reported here the plot of this first spatial pattern, and as you can see, there is indeed an asymmetry between the more affected and less affected part. And this represents the loadings of for each region. The second component was related instead to the disease-induced gradient. We coded ventral striatum that are relatively spared compared to putamen. And the last component represents the progressive loss in the less affected striatum, which also correlated significantly with disease duration. An interesting thing of this is that these three components are orthogonal. That means they are independent. So the author suggests that this may mean that the underlying mechanism behind these may be independent. Instead, moving to the unique pattern of the two radio tracer, for the DTBZ, the pattern was similar to the first canonical component, so an asymmetry between the less and more affected striatum, and this may suggest that this radio tracer is more sensitive to the direct dopaminergic terminal degeneration, while the DAT was more linked to compensatory response to the more severe dopaminergic loss detected by DTBZ. So to summarize also this study with the two main points, we saw that this approach provided not only localized alteration, but also the spatial extent of such alteration, emphasizing that the brain is better understood in terms of network behavior. And they were also able to decompose, share the common information between two different radio tracers. And these components were also sensitive to the disease discrimination and progression. Moving to the third and last case study, in this case, the main target was the glutamate system, and we have a work from Dubois et al. Of 2023. In this case, the cohort under investigation was composed by 33 healthy control and 17 patients with focal cortical dysplasia. That is a form of epilepsy that it is linked to cortical malformation and the drug-resistant seizure. And they were scanned to measure the metabotropic glutamate receptor type 5, that is a postsynaptic G protein that may be linked to epileptogenesis of this disease. And the aim of this work was to characterize this mGlur5 network in patients with this focal epilepsy through graph theoretical analysis. And this study is particularly interesting because they applied an individual level approach. And this approach consisted in the first extracting the binding potential from the cortical region based on the parcellation scheme that in this case was the Glasser. And then they used the voxel of each region to construct the probability density function using kernel density estimation approaches. And then they evaluated the pairwise node probability density function similarity using a Jensen-Shannon divergence that is a measure similar to an entropy or a mutual information measure that quantify the dissimilarity between two probability density distribution. Then they converted this measure to a similarity measure bounded between 0 and 1. And this was performed at the single subject level, so they obtained one matrix for each subject. Moving to the statistical analysis, they used different network metrics like the small world index, global and local efficiency, resilience, and participation coefficients. Where participation coefficient is a measure of the tendency of a node to being part of more than one community. So it's a way to quantify how important a node is in a network. And then they also calculated the resilience, that is the robustness of a network when you delete one or more node. And they performed both random attack to remove node at random and a targeted attack removing node with high participation coefficients. Then they compared this graph metric between the two groups using area under the curve, also non-parametric test. In this case, they found that the patients exhibited a more regularized network with a reduced network efficiency and decreased small worldness. And this suggested that the patient may have a less integrated network. The author suggests that this may be to network reorganization or disease duration, or seizure frequency. Moreover, the patients also showed a specific vulnerability to the deletion of high participation coefficient node with respect to random graph with random attack. And also the patient showed an association between the disease duration and the local efficiency of the hub node contralateral to the lesion. And this suggests that the seizure activity may reduce the efficiency of transmission information in distant regions that are essential for network integration. So these results, for the author suggests that this result support the idea that this disease is better understood in terms of network and system-wide disorder instead of considering the region as individual regions. This was the last case study. Now I will go through some other interesting results from other paper included in the review, and then I will conclude with a general discussion. So I will start with a study on the dopamine system. We have a study of Sanchez, Catasus et al. And this is an interesting study because they combined tractography with dopaminergic PET information, and they were able to show an imbalance between acetylcholine and dopamine in the striatum during the early stages of Parkinson's disease. So this was interesting because they were able to combine multiple modalities. While for the serotonin system, we have another study of Fu et al. Of 2018. And in this case, using SSMPCA, they identified disease-induced alteration of the serotonergic system in PD patients that was also linked to abnormality in dopaminergic system, creating a link between these two different neurotransmission systems. While moving to the muscarinic system, the study of Colloby et al. Of 2021 were able to identify spatial covariance pattern in M1 and M4 receptor that were able both to distinguish subject with dementia with Lewy body from healthy control, and also these patterns were positively associated with cognition and neuropsychiatric symptoms following pharmacological treatment. So they also found a bridge between these network measures and clinical scores. Finally, regarding the opioid system, we have a study of Ashok et al. Of 2019 that through the correlation analysis revealed a global increase in mu receptor connection strength in subject with schizophrenia. And the last study of Fang et al. Used an independent component analysis to link resting state network of functional MR with source network obtained with UCBJ that measure the synaptic density. So they were able to find a link between a possible link between synaptic density network and functional connectivity network, again creating a bridge between two different modalities. So to conclude, through this review, we were able to see that molecular connectivity can effectively integrate both localized information and broader network level relationship, enhancing the robustness of the classic traditional univariate analysis. And through these approaches, the authors were also able to uncover a disease-related alteration, revealing that the brain is indeed a network, and this system-level analytical approach better fit the purpose. And we saw also that the molecular connectivity is particularly valuable in the study of neurodegenerative disease, as we saw, for example, in the Parkinson's disease. And this may be linked also to the fact that some diseases like these are better explained also in terms of group level analysis, while other type of disease, like for example psychiatric disease that are more heterogeneous, or other focal pathologies necessitate an individual level approach. And finally, collectively, these findings showed that the molecular connectivity is a robust framework for integrating not only multiple radio tracer, but also multi-modality. But there are also some limitations. And one of these is a standard terminology is still lacking. And this is a problem principally for a communication between different studies, and also between different, within different community, for example, with the fMRI community. Moreover, although statistical validation is commonly employed, the absence of corroboration with pharmacological intervention or ex vivo data limits the interpretability. So for sure, in future, the research study should prioritize evaluating changes after treatment or therapeutic agents. Also, the scarcity of longitudinal study restricts the ability to assess the effectiveness of molecular connectivity approaches in tracking the dynamic network. And finally, the majority of the analysis rely heavily on group level analysis. And as we saw, this may be a good approximation for some disease like Parkinson's disease, but it's not the best approach when we want to analyze a heterogeneous disease or focalized disease. So if you are interested in this work and you want to know more about it, you can find the paper on imaging neuroscience. And to conclude, thank you, Mattia, my supervisor, all the lab, and all the co-authors, and all the molecular connectivity working group for this work. And thank you for your attention. I don't know if Mattia wants to add something.
I wait for the questions. Thanks, Mario.
Okay, I've concluded with the talk.
Now is your time. Who wants to start?
Sorry, I have a very simple practical question about the results you displayed in the early third when you looked at the graph theory differences. You know, I think it was transitivity and global efficiency, maybe. Can you go back to these graphs? Sorry, I was struggling in my mind to Yeah, there it is. Yeah, great. By the way, I thought that was very nice. Also with the non-parametric permutation stuff. So here, can you just? I was puzzling the stuff on the x-axis. What is that? Is that a sample size or a binarization threshold? Or sorry, I was somehow I couldn't get over that. I was thinking about it, you know, three slides later. Still.
Is the binary is the threshold because they
okay,
matches with different density value.
Oh, okay. So you show there's some nice invariance with regard to threshold, and it isn't really crucially dependent on how you binarize. Okay.
I mean, one thing that is remarkable by reading these studies, like they all use different methods. Also, when they claim to use the same, they were, you know, different settings. They were adapted to the particular context of the problem where they were applying these methods to. It was very difficult to try to get a summary when you have such a different methodology. Some of them were, you know, statistical analysis at the group level, looking at distributions of these connections. Other, they went up to analyze network metrics. Other were individual metrics. Other were regularization, non-regularization. So extremely difficult to find, you know, there isn't one method that works for all. That's for sure what emerged from these discussions. It is possible there might not be one because, you know, you can create these statistical objects in different ways and use that for different purposes. I can see Igor and Christine, so I don't know who raised the hands first. Christine, do you want? Yeah, Igor. Okay, Igor, you raised the hand first, please.
My turn. Yeah, thanks a lot, Mario. Fantastic work. I mean, if you think back that you started with over 7,000 studies, great, thank you very much. Mario, all studies are based on inter-subject analysis, right? So it's across subject statistics.
Yes, the majority of studies are inter-subject. If I'm not wrong, there are maybe two studies that are also intra-subject.
Yeah, because I remember Matthias' comment in Munich that there are now approaches that allow to study or to apply neurotransmission PET also dynamically. So potentially also allowing to study molecular connectivity within a subject, right? Were there such studies in this review or only across subjects?
Do you want to answer, Mario, or you want me to answer?
As you prefer.
You go ahead and then I might add to your question, to your answer.
I don't know if we are talking about functional PET or something like this.
Yeah, there is also no unified terminology also for this method, but I mean a PET within one subject, where you potentially also could calculate some statistics indicating connectivity, but within one subject.
Yes.
So not across subjects.
Okay. For example, in this case, with this approach that you can obtain the matrices at the individual level. So in this case, you could potentially compare the individual subject in terms of molecular connectivity in time. But regarding the functional PET part, those studies are only with FDG PET. There is one study with functional PET with FDOPA, but is a traditional univariate analysis approach. So we don't have the functional PET study with the neurotransmission tracers yet.
And to add to your question, Nico, yes, there are methods to build these matrices at individual level. Some of them use the time activity curve, so there are the dynamic of the tracer over time. Others use features extracted from tracer kinetics in specific regions. There are perturbation covariance analysis where you basically look at deviations of your individuals out of samples that define a reference cohort. So there are others, and as a matter of fact, with Mario, we recently investigated the use of one of these methodologies applied to fluorodopa and compare cohort in schizophrenia and in Parkinson's disease as a clinical validation models. And we did find that the methods is capable of differentiating the disorders and the different characteristics of the disorders, but also to be informative of drug medication response, in particular classifying responders, patients with schizophrenia responding to standard treatment as compared to non-responders or resistant patients. And also, we found with this method that you can look at the different course of the disease. So we started from healthy controls, individual high risk of schizophrenia, first episode of psychosis patients, and chronic schizophrenia, and we found different pattern of connectivity. Now the problem is like we did not have a longitudinal sample to look at these deviations over time. That will be, the final evidences and test to evaluate how these methods can track disease progressions. But I guess, we are getting there. So the more these tools are known, the more you will see people apply them. Also, in terms of numbers of publications, is growing every year in terms of papers and applications. So if they not be already applied, I'm expecting something coming out in the next months or in the next year.
Understood correctly, you see a bright future for functional PET in the field of neurotransmission too, so beyond FDG.
Yes, for sure. I mean, for the studies I'm working on, I'm systematically using these methods to complement traditional analysis. This is very good and important. This is not going to replace traditional analysis. This is complement traditional analysis, especially when you have models of disorders that there is an interplay of regions and an interplay of systems, and these are good to put things together. They works better if you have an hypothesis, because you can have a lot of information. Look at these matrices over here. There are hundreds of thousands of points. So going completely data driven to me is a risk. But if you have a model of the disease, you have an idea, certain hypothesis, with these elements you can test them. And I think that's very important. I don't know if Christine will agree with me on this, since you have been in the field so long enough to say if this is valuable or not. Christine, can you hear me?
I can. Sorry, it's that sort of leads on to my question. I wasn't sure if the previous person had finished. I'm so sorry. Okay. Is that all? Shall I continue?
Yes, please.
Okay. Well, it kind of feeds into my question, but I have a couple of questions. Okay. I think this is really important. And obviously it's focused on PET, this presentation. So I guess one of the points I wanted to make was, how do you think this sort of like the molecular imaging connectivity could feed into, say, MR methods as well? Because obviously we've got more resolution with the MR methods, but they give different information. But, tapping into how the molecular connectivity of PET or SPECT can also connect with the MR approaches to kind of give more information. It's just a mushrooming area, as I see molecular connectivity, and really important. So thank you for this presentation. It's really great to see this sort of work being presented, because it's something that's, individual brain regions don't sit in silo, they connect with each other. And so talking about how it interacts with other imaging and maybe non-imaging methods, but again, that's perhaps a topic for somewhere else. What do you think is needed to start standardizing these approaches across labs? Because it's all very nice it's happening here, but it could be happening slightly differently in other labs, and somewhere along the line you want to be able to put these data sets together so you know, standardization in molecular imaging isn't always an easy thing to do. I know that coming from a pharmacology background. So I just wondered, like, you know, has it already started? Because I'm a little bit perhaps maybe behind on this, or is it something that needs to grow in the future? So just like to get your thoughts on all that. And I'm really sorry if that was slightly disjointed, but I think this is a very exciting area. And I think this is where molecular imaging will move to, because for some diseases like ALS, Parkinson's, it's not about individual brain regions. It's about the brain as a whole and then how it connects with the rest of the body as a pure biochemical, physiological sort of like system. So yeah, I'll let you answer all of that. Thank you, Mattia.
You're welcome. I try to be brief and leave the stage to Mario as well. And you know that for me is a problem. So we already started applying some of these methodologies to a cell data from MRI. Because at the end of the day, you know, we could assume them to be molecular maps of cerebral blood flow. And we applied those in some studies in schizophrenia, and we found some findings of that were not captured or were complementing the standard analysis of changes in CBF. So for those, we started working on this. Crazy enough, we have been working on applying these methods with TSPO. And I know it's not exactly neurotransmissions. I mean, we pretend to be neuroinflammations or TSPO, whatever is labeling. But we found extremely good results in terms of fingerprinting. So we can capture somehow individual characteristics that with traditional analysis is very difficult to see. But also, we have been able to identify some statistical property that with standard TSPO analysis we are not able to get. You know, all this variability and this, you know, poor differences across cohorts. And this is not just about TSPO. Is when we apply to this, we also applied some Parkinson data with serotonin scan and synaptic density, a data set that you should be aware of. And I have some preliminary results, and we found stronger evidences of progression, disease progression, as compared to the standard analysis. So I can see the value. I can see that it can be generalized across different modalities. Some people have applied this to transcriptomic maps and compare transcriptomic maps to imaging maps. So, you know, I guess creativity is quite large. And I know Andreas Hans is working with pioneering the functional PET for the FDOPA. I'm sure, you know, he has other things in pipeline. So the functional PET is really a terrific and very nice approach and a good one. It's difficult to implement. I guess we need the PET center to engage with these methodologies because the protocol is different. But I guess, you know, we can start validation of these methods by simply analyzing the data that we already have. And in terms of this data, I think the valuable data will be data with blocking agents, test, retest. So there where we have control challenges that, you know, will tell us whether we are going to the right direction or not. So for the validations, I think we just need to keep working on this using data sets that, you know, give us the possibility to answer to specific questions. I'm a bit, at the moment, all the clinical applications that we are doing, they are more to use the disease as a model rather than to use to find something on the disease, because there is so much unknown in these methodologies that, you know, when it gets too much, it gets confusing. That's my, so I will see more on this, not just because we have the chance, but because the results are interesting and intriguing. I don't know, Mario, if you want to add more to this answer.
Just only one thing, that if you want to apply this approach as a data-driven approach, you can apply it to whatever data you want. But if you want to have value from it, I think that, as Mattia said, you need strong a priori hypothesis before applying these network approaches.
I mean, do you think the best sources of data to kind of sort of validate these models are kind of longitudinal data sets where you actually look at disease progression within subjects and then within a cohort of the same subjects to kind of really hone out molecular connectivity at point 0, you know, whatever time you come in on the disease or healthy, and then with disease progression to see how to kind of get a feeling for how the connectivity changes with disease progression or with therapeutic intervention, how it goes in the reverse, potentially, depending on what's being targeted. I mean, is that kind of where you think this needs to start, or do you think that's already being done?
It's not been done. So that's, I think, where we want to go. So I think for human applications. For the validations, I think the animal models here where we could, for example, play with focal or like specific lesions where you can break some certain circuits and see how that will affect the statistical object, that would be very important to prove the performances. Why I think the value will be definitely linked in longitudinal progressions. Also, in the precision medicine approach, I think we can capture things of the individual, of the patients, that will probably be good indication of which treatment to go. And I can see these things already done with fMRI. I'm thinking, for example, this fingerprint, drug fingerprinting that Ayana Cash is doing with all the drugs, in animal works. Same thing could be applied to humans. And we just need to pull data together, probably get the best out of what we already have, and then start to planning for, more specific experiments in order to test what is missing.
Okay. Thank you.
You're welcome.
Okay. I don't know if there are other comments. I have three general considerations that I want to point out. Three different levels. The first one is on molecular connectivity, more general, because I think that while is very intuitive speak about spatial patterns on patients, when you move on statistical metrics that you mentioned, like small word index or other more complex, the translatability of this approach in a clinical context become more difficult. And this is what is happening with the radiomics, for example, where statistical approach for heterogeneity of lesions are applied, but are difficult to translate in a clinical context. The second consideration is on neural transmission and on specificity of this radio tracer, because as you mentioned it before, there are different actors in synaptic transmission. So I don't know if there are radio tracers specific for glial transmission, for example, because there are receptors that are specific for glial cells, and there are a lot of theory of gliocentric theory for disease. So probably this can be a point to reflect. And the other one is to link to Christine consideration is about the role of PET connectivity compared to other metrics, because and for validation, because as you know, there are Allen maps for transcriptomics that you can compare neurotransmitter distribution with PET distribution. Or for example, there are several works now of Calamante that with PET informed neurotransmission to combine PET and DTI to build your matrix. So what do you think that is the place for molecular connectivity in neurotransmission? Thank you.
All of them and potentially none of them. I don't know. It's very early days, Carlo, to say what would be the place for these methods. To be honest, I agree with you. I don't like too much to push on the network measures because you lose clinical interpretations. For example, with Mario, we found a very simple approach that the perturbation covariance analysis has with these methods is to look for deviation scores. So something that would reflect how much the pattern deviates from normality, and this could be a generic measure, so of something going on differently in that patient as compared to the others. At the end of the day, these are simplified measures of trying to summarize are complex objects, but then when you want to go to clinical practice, more to clinical translations, that's the thing that you have to do. You're trying to simplify the system to get one or two numbers out of them. And for the rest, I think we just need to have good people with good questions and get out of these tools the right things that they can provide. So complementing MRI analysis, complementing other modalities, complementing electrophysiology, complementing the knowledge that we have of the brain. So you mentioned glial cells. So if you have radio tracers for these functions, we can combine them together. Now there are more studies nowadays where you have more than one tracer in the same individuals. So trying to tease apart sub-affinities, for example, or different components. There are studies now looking at TSPO and astrocyte markers to try to look at the two sources of inflammations that you have in the brain, just to make an example out of neurotransmissions. But I think it's all the sub-affinities that this radio tracer will have. So these are important questions. We just have to use this as a tool, but start from the questions. That's what is very important. Is always the case.
Thank you. I don't know if there are other comments in chat, nothing. So I think that we are also in time.
So you don't blame the Italians, okay? Our times is just 5 past, so it's okay within Italian etiquette to be 5 minutes late. So in Italy, you know when the meeting starts, but you don't know when the meeting finishes. So it's actually surprising, but that's I've learned coming back.
Fortunately, there are limits on
Zoom.
Zoom, yes. So
okay, everyone. Yeah.
I have seen that Sin have remembered that for the next meeting of Simon Eikhoff that we will have, there is a link for the next meeting. And if there are no other questions, I would again thank you, Molecular Connectivity Working Group, for this opportunity, and speakers, and about 50 participants in the peak. So thank you again for your presentation, and we'll see the next time.
Thanks
Thank you.
you all.
Thank
Have a
you.
good weekend.